here. Geoff, of course, is at Duke University now, but he has a pretty storied genealogy coming up through Yale, Johns Hopkins to do his MD, PhD, and then on to Stanford to do fellowships and postdoctoral fellowships. Subsequently, assistant professor at Columbia University, and then moving to Duke, near neighbour, where he is currently professor of medicine, the director of the Ion Channel Research Unit, and also the interim director of the Cardiovascular Research Centre there. And so, Geoff has had sort of many important papers over the years. His particular interest is, of course, ion channels, major interest in calcium channels and the proteins that associate with them, Calmodulin, various other accessory subunits of ion channels. And so, I'm very much looking forward to his talk today on the dysregulation of ion channels and disease processes.
So thank you, Rob. Can everyone hear me okay? My little fighter pilot microphone is working okay? I don't know if everyone can see what's going on with the slides, then I'm okay with it. Just get my glasses all set here. So it's a real pleasure to be here. I had a fun time talking to people at dinner last night and realized that it's really kind of a mixed cardiovascular and neuroscience audience. So I kind of put together a talk that tries to hit on both, and hopefully it'll go under the time frame. So my lab is interested in channelopathy. So these are disorders of ion channels, and channels are expressed in various tissues, and so when they're dysfunctional can cause disease processes in a number of tissues within the body. So we're right now working on almost all of these different areas. I'm going to focus specifically on some of our work in the brain, and as a cardiologist, I'm particularly interested in the heart. So just as a background, as most of you know, ion channels regulate action potentials in excitable cells. So as a cardiologist, I'm interested in cardiac myocytes, and so there are a bunch of proteins that form the pore-forming subunits for the inward depolarizing currents, such as the calcium channels that Rob mentioned, and sodium channels, which will be a major focus of this talk. And then there were outward repolarizing currents, such as the potassium channels. We now know a lot more about ion channels in that they're not kind of these single proteins that form pores, but there are these large macromolecular complexes of proteins that bind to and regulate the channels. And so this is a really nice study that's almost 5 years old now that looked at the calcium channel macromolecular complex from brain. And so if you focus down, there is the pore-forming subunit, which is shown in red, and then there are these accessory subunits, which I call the traditional subunits, and these are usually the beta or gamma subunits. They come from when people like Bill Catterall were purifying proteins over 20 years ago now, and so these are the ones that are most tightly associated and sometimes absolutely required for function. But now with other types of methods to investigate channels and proteins in general, we now know that there is a much wider degree of proteins that are modulating these proteins. And these I call the next generation chips, or channel interacting proteins. And these can have a number of properties. So they can be very dynamic. They can be interacting with multiple channels. So one particular auxiliary subunit can do multiple things. And they're important in disease is highlighted by the fact when you think about certain cardiac arrhythmias, such as long QT syndrome. This is a congenital disorder that leads to life-threatening arrhythmias. And you look at the first three most common loci. They are the pore-forming subunit for 2 potassium channels and 1 sodium channel. But if you look at all of the other loci that have been recently identified, highlighted in red here are these channel interacting proteins. So you can get the same end phenotype or disease, whether you have a mutation in the pore-forming subunit itself or in some of these auxiliary proteins. So as I mentioned, this next generation chips, this part of these large macromolecular complexes, can be multimodal. For example, there are a set of proteins called K-chips, but they were originally identified as potassium channel interacting proteins. And it turns out that we and others have shown that they do other things, such as regulate calcium channels. And then you can have more general proteins such as Calmodulin, which is a protein that my lab has spent a lot of time working on. And this is the ubiquitous kind of translator of calcium into cellular response. And it turns out that these Calmodulin and Calmodulin-like molecules are also regulators of various ion channels. And so if you look, for example, in Brugada syndrome, which is another type of an inherited arrhythmia, once again, you can see that the most common locus appears to be the pore-forming subunit of the sodium channel, but in red are all of these other accessory proteins or chips that can also cause disease when mutated. And I'm going to talk to you a little bit about some of our work with a particular set of these proteins called fibroblast growth factor homologous factors. So this is a long name, so I'm just going to call them FHFs. And so we recently showed that FGF12, which is one of these FHFs, which I'll explain to you in a moment, is a locus for Brugada syndrome. So this is, as I said, the kind of a summary of what these next generation chips will do, and what I'd like to talk to you about today is some of our work focusing specifically on these FHFs and also on Calmodulin, how they modulate ion channels and how they, when dysregulated, can contribute to disease. So let me introduce this FHF family. So these are 4 of the canonical fibroblast growth factors. It's FGF11 through FGF14. They got their name because they have a canonical core domain that's very homologous to the FGF superfamily. They have extensive N-terminal splicing, and then they have a non-canonical extended C-terminus. All of these N-terminal splice variants for the 4 different genes don't have a signal sequence. So these proteins never get secreted, and so they never actually function as growth factors. And so some people call these the IFGFs for intracellular FGFs, and then the name that seems to stick right now are these fibroblast growth factor homologous factors, these FHFs. And so the mystery of what they did, since they weren't functioning as growth factors, and by the way, if you make recombinant protein and you pour it onto a cell with an FGF receptor, it won't actually engage the receptor and activate the receptor. And we know structurally now subtle changes that cause that to happen. So the mystery of what they do began to unfold when people discovered that they interact with the C-terminus of voltage-gated sodium channels. And so here is a voltage-gated sodium channel, outside, inside, and there are the 4 sets of 6 transmembrane domains, and FHFs bind to a conserved sequence in the C-terminus of the pore-forming alpha subunit. And this is a crystal structure that we did a few years ago showing the C-terminus right here in brown with the 2 proteins that I'm going to talk to you about today, Calmodulin bound to this long alpha helix and the FHF bound to this more globular domain, more close to the transmembrane domain of the sodium channel. So these seem to be important. As I already mentioned, and I'm going to talk to you a little bit about, we've identified one of these as a locus for Brugada syndrome. They're also mutated in several different neurological disorders. The one that's probably the most well characterized, although I'll try to explain to you that I think that the disease mechanism is a little bit unclear, is spinocerebellar ataxia 27. So this is a neurodegenerative disorder of the cerebellum, and you can tell that there are a lot of them because this is spinocerebellar ataxia 27. And it turns out that FGF14, one of these non-canonical FHFs, is the locus for this particular disease. And FGF13 has also been linked to several neurological disorders. And so what I want to start to talk to you about is some of our work about FGF12 being a candidate locus for Brugada syndrome. So we got into this because I showed you the crystal structure, which I'll come back to towards the end. We were very interested in understanding how Calmodulin interacted with the sodium channel, and through our kind of structural forays, we started working on this FHF protein. And while we were doing that, we went back and looked at some of the original data. And so this is a in situ hybridization that Jeremy Nathans performed. He was the one who originally isolated these proteins. And so this is FGF13 or FHF2, and you can see this in situ hybridization. You can see it in the CNS, and so a lot of people have been studying the roles of these proteins within the CNS. You know, as a cardiologist, I saw that there's a big red blob right here, and so I said these must have some also important functions in the heart, and so we figured that we would begin to look at them. So we developed a knockdown strategy where we used adenoviruses that we could then infect adult cardiovascular myocytes and then query the function of the sodium channels, because we knew that they interacted with sodium channels in the absence of these FHFs. And so here's the general system, and this is published, so I'll go over it relatively quickly. So we have viruses that will identify transfected cells because they're green, and then those that also have and then we can also detect with a custom antibody, the FGF13. And so here's a scrambled virus, and you can see that the FGF13 is still present. And then when we knock it down, you can see that most of the FGF13 has disappeared. And so what are the consequences for sodium channels? Remember, this is a sodium channel binding protein, one of these sodium channel chips. And so this is a set of current traces. So for the few of you who might not be interested in ion channels or used to looking at ion channel traces, suffice it to say that the more it goes down, the bigger the sodium currents. And so you can see that after knockdown with the shRNA, it's just smaller, so there's less sodium current. And so why is there less sodium current? Well, it turns out that there's probably at least two mechanisms. So this is an assay where we're looking at the availability of the sodium channels at any particular voltage. And so you can see that after knockdown, you get this shift to the left. So that means at any particular voltage, less sodium channels are available. So it's changing the voltage dependence of the channels. And then in addition, when we looked at what was going on with the protein, we found that there was less protein, less sodium channels getting to the cell surface after we had knocked down the FHF. So here's the total amount of protein with an antibody against the sodium channel. And here is the membrane fraction, which we isolated through a biotinylation process. And you can see that after knockdown is that you have less protein, and this is quantified here. So we got about a 50% reduction in the amount of sodium channel that's at the cell surface without any change in the total protein. So the consequence is that you have less channels available at any voltage, and you have less total sodium channels at the membrane, and so you get less current. And so, as I mentioned before, the most common locus for Brugada syndrome is the sodium channel. Most of the mutations are loss of function mutations in the sodium channel, so we thought that the FHFs could be a locus for Brugada syndrome. So then we went back and looked at human tissue and determined that FGF12, I was focusing before on FGF13 because that's the most common FHF in rodent cardiac myocytes. FGF12 is the most common FHF in human adult heart. And so then we went and we queried a database that was available from Mike Ackerman and Peter Schwartz of patients that had Brugada syndrome but did not have any mutations in any of the known genetic loci. And we asked specifically whether we could find mutations in FGF12. And sure enough, we found a single patient in about the first 100 that we queried that had this mutation in this very conserved glutamine at position 7. And you can see that it's highly conserved across species. And so this was immediately interesting to us because this now is in green is the C-terminus of the sodium channel. Purple will be the end of the talk about Calmodulin. The FHF is in yellow. If I kind of break them apart and splay them open, you can see that this glutamine is right over here, and this was of immediate interest to us because we had previously identified that there was a particular lysine that sits right here and forms a salt bridge with this aspartate residue over here. This is actually quite unusual, and I'm going to show you that interaction is very important. It's quite unusual because usually the important interactions are not at the kind of the extra outside of the interface, but right in the middle. Yet we were able to show that this salt bridge was really important. So this is using isothermal titration calorimetry, which is a relatively accurate way to get affinities. And so here is a wild-type pair of the C-terminal domain of the sodium channel and the FHF. And then we can essentially do a charge swap. So we can make this lysine into a negatively charged glutamate, this glutamate into a positively charged lysine, and look at a mutation of one, or swap them and look at the mutation of both of them, and see whether we can change the affinity and restore the interaction. And so here is about 100 nanomolar affinity for the wild-type pair, and when we mutate just the C-terminus, turn this into the lysine, you can see that we get a marked increase in the affinity. If we do the same thing by just mutating on the FHF side, also a marked increase in the affinity. But then if we mutate both, so we've completely reversed the the charge, then we can essentially restore the affinity back towards the wild type levels. So that in the patient, that residue right next to this critical interacting residue we thought was very important, and so we repeated those experiments. And so here's the wild type, once again about 100 nanomolar affinity. Here's the mutant, and it's about a threefold difference in affinity. And we're able to show by immunoprecipitation that that caused about a 50% reduction in the amount of protein that we're able to pull down. And so then we wanted to know what the consequences of that were. So we've been studying these for a while, and we realized that looking at heterologous expression systems was challenging because, as I said before, these channels are large macromolecular complexes. And so if you stick your favorite ion channel and your favorite channel interacting protein in a hex cell or in a xenopus oocyte, sometimes you might not get accurate information about what the channel and what these channel interacting proteins did. So we wanted to go back into a cardiac myocyte to explore what the functions are in the native environment with all the endogenous proteins around. And so we did a kind of a knockdown and replacement strategy. And so here's what we did is we knocked down the endogenous FGF13, which I showed you was the main FHF in rat adult ventricular cardiac myocytes, and then used another virus to express the FGF12, the human version, and we either express the wild type or the mutant. And so we added a his tag so we can detect that the mutant protein was present, or the human protein was present. And so under these systems, then we could then query what the consequences of this mutation, which reduces the affinity by about threefold, what it does to sodium currents. So here are the control myocytes. Here's the FGF13 knockdown, and as before, we get about a 50% reduction in current. When we replace with the wild type FGF12, the human one, we get a restoration of the current. So this is knockdown plus replacement. If anything, it's even better. And you can see that the mutant is unable to restore the knockdown. So control to knockdown, restoration with the mutant, it doesn't work. Here are the summary data here. And it suggests that this threefold reduction in affinity is therefore enough to affect the sodium currents. And so this mutation is essentially the equivalent of a loss of sodium channel function. And we think that's why this particular patient had Brugada syndrome. So in other work that I'm not going to have time to go over today, here is a summary of what we've been able to show is that FGF13 binds to the intracellular C-terminus of the voltage-gated sodium channels. It's involved in getting the proteins to the cell surface, and it's also involved in regulating those channels that are at the cell surface in terms of their kinetics. And we were able to show, using some monolayer experiments, that the loss of the FHF is somewhat equivalent to the loss of the sodium channels, and they got a reduction in current in conduction velocity. So let me switch gears and go into the brain to try to appeal to some of the yes?
Is the mutant that changes the sodium channel gating or just the traffic?
So it does both. It changes, you get a reduction in the amount of sodium channels at the cell surface. So it's just like the knockdown. And we also see this shift in steady state and activation as well. So we think that that reduction in the affinity is essentially causing less interaction, and so it's not quite like a null, but it's a very weak interactor. So we see both of those processes. So let me switch to one of the neuronal FHFs. So I mentioned before that FGF 14 is the locus for this neurodegenerative disorder called spinocerebellar ataxia 27. This is a disorder of the cerebellum. The cerebellum has a very interesting circuit with the final output of the cerebellum are these Purkinje neurons that are shown here, and there's a particular synapse between the granule cells of the cerebellum and through these parallel fibers onto the Purkinje neurons. And FGF14 is expressed both in granule cells and in Purkinje neurons. And so we decided to look specifically at this synapse, this granule cell to Purkinje neuron synapse. Now, cerebellum is interesting because if you essentially take a slice of cerebellum and you start recording and you go off to lunch, I mean, they just kind of fire forever. So unlike cardiac myocytes, these channels don't go into an absorbed, deep inactivated state, and so it allows them to keep on firing, and that's very important for cerebellar physiology. And if you look at what happens to the FGF14 knockout, you can see these are actually stimulated action potentials, but you can see that there's markedly less action potentials in the FGF14 knockouts. And so what was interesting to us is, and this is not our work, this is the work from David Ornitz, who's a big FGF person at WashU, has done some really exciting studies on this. But what was interesting when we started looking at this is, I told you before that when we knocked out FGF13 in the heart, we saw a reduction in the amount of sodium current and a reduction in the number of sodium channels at the cell surface. So it turns out that in cerebellar granule cells and Purkinje neurons, that same process doesn't happen. Granule cells or Purkinje, it doesn't really matter because it's the same in both. I don't remember which one I'm showing the data for right now, but you can see that after knockdown that you have no change in the current density. We were able to show that it definitely interacts with the channel just like it does in the heart, but there's no change in current density. So it suggested to us that the disease mechanism had to be different in cerebellum versus in the heart with the Brugada syndrome. And so we started looking carefully at the kinetics of the sodium channels in cerebellum after we knocked down FGF14. And excuse me, so these are a bunch of different kinetic analyses. And so the first thing that we see is after knockdown is we get the shift in the steady state inactivation, as I showed you before, but we also saw some very interesting changes in other aspects of the kinetics. So you can see, for example, that the after knockdown is you have faster, more rapid, and more complete inactivation. And I mentioned before that the cerebellar neurons have to continue to fire, and so their sodium channels have to function differently than in cardiac myocytes in that they can't inactivate and go into that deep absorbed inactivated state, or otherwise you wouldn't be able to have that continued firing. And so what we noticed after knockdown is that we got a reduction in what's called the late current. So you get this transient opening of the channels, and then there are a small portion of channels that continue to stay open. So this is what's called late current. In cardiac myocytes, this is really important because mutations that increase the late current lead to after depolarizations and lead to arrhythmias such as long QT syndrome. In cerebellar Purkinje neurons, you actually want to have a lot of late current because that means that the channels are not inactivated and then they continue to fire as they're supposed to do. So it's very interesting that when we saw that we could see this reduction in late current, we also showed that the kinetics of inactivation were also faster. And let me also point out on this slide is that in all cases, we've knocked down and then rescued with an shRNA insensitive version, and you can see that in every case, we always restore it back to the control levels. So we know that our shRNA is on target, and it's not causing any sort of other problems in the cells. So seeing this kind of bias towards the inactivated state made us think about something called resurgent current. So in cerebellar Purkinje neurons, particularly, it's thought to be NAV16 or SCN8A is a particular neuron that undergoes this property called resurgent current, originally described by Indira Raman and Bruce Bean. And what happens is that you have the initial transient activation, and then you have this, at least compared to cardiac myocytes, you have this increased late current. And then if you repolarize the cells, but still at a potential that's permissive for channel opening, you see what's called the resurgent current. This is particular to certain neurons, particularly in the cerebellum, because you can see if you isolate neurons from the hippocampus that you don't have this process. And so the molecular mechanisms of this have not been very clear. The idea is that you have an inactivation particle, which is thought on the sodium channel to be the 3,4 intracellular linker that essentially forms like the hinged lid on a can and closes the channel. But in cerebellar Purkinje neurons and granule neurons, you have this blocking particle, which some people believe is the C-terminus of one of the beta subunits. And this essentially gets in the way of the inactivation particle. So upon repolarization, the inactivation particle is not there, the blocking particle is removed, and you get this resurgent current. And then that sets up that next action potential, so you can have that repetitive firing. So, as I said, the mechanism for that was really not that clear. So when we saw after knockdown of FGF14 that we were biasing these cells towards the inactivated state, we thought maybe we would be interfering with this resurgent current and therefore causing more inactivation and therefore giving the phenotype that I showed you on the first slide with the FGF14 knockout, which would explain the disease phenotype. And so here are some of the data specifically investigating that. So this is looking at the resurgent current, and this is after knockdown. You can see that it's markedly reduced. Once again, it's rescued when we express an shRNA insensitive version, and this is some of that summary data as well. So it looks like the interaction of FGF14 with the sodium channel is essential for this resurgent current, and therefore the loss of this in the disease process could be what's pathogenic. Now, having the crystal structure, we could then ask questions such as whether the direct interaction of FGF14 with the sodium channel was essential. So this is a different mutation. This is one that we actually designed. So we knew that there was this arginine residue right here that kind of has a finger that sticks into this deep crevice on the sodium channel side. And so we can mutate this arginine to an alanine, so it leaves this structure essentially intact, but it no longer allows interaction with the sodium channel. And this is some of that isothermal titration calorimetry data that I showed you before. You can see nice affinity with the wild type pair, and then when we make this single alanine mutation, you essentially see no interaction whatsoever. So now, and this just shows you that we no longer see interaction by immunoprecipitation. So now we can use this knockdown and rescue approach, knock down the endogenous FGF14, rescue with an shRNA insensitive FGF14 that has this single point mutation that renders it unable to interact, and ask what the consequences are. And so here is the knockdown, here is the rescue with wild type, and then when we rescue with the mutant that can't interact, here's the rescue with the, I can't see from this slide, but when we rescue with this R to A mutation that can no longer interact, you can see that there is, we don't get the restoration of the resurgent current. And here are the summary data right here. So it suggests that that interaction is absolutely critical for its function. And then going back to querying the action potentials, so here's summary data from a bunch of different mutations we made. So here is the control scrambled shRNA. Here is the knockdown at two different current injections. You can see that you get less action potential. Here's our ability to rescue it with the shRNA insensitive wild type. And here is the R to A mutation as I showed before, and here's a different mutation that I won't go over today that allowed us to figure out exactly how these two proteins are interacting. So it looks like the disease process for spinocerebellar ataxia 27 is not because of loss of sodium channels, because as I showed you at the beginning, the sodium channels are unaffected after FGF14 knockout or knockdown in the cerebellum, but instead it's a bias towards the inactivated state, and so you lose the repetitive firing that's essential in the cerebellum. So Juan Pablo is a graduate student in the lab who has been interested in looking at other roles for these FHFs in neurons. And so he started by looking at immunohistochemistry of the hippocampus, looking at pyramidal neurons, because both FGF13 and FGF14 are both present in the pyramidal neurons. And so we thought that this was interesting, that you'd have these two different FHFs, both of which have the capability of binding sodium channels, and do they have distinct functions? And one of the first things that he noticed was that they both localize to the axon initial segment, which is the area in neurons in which sodium channels congregate and cluster so that they could fire action potentials efficiently. So they're there where they should be with the sodium channels. But within the axon initial segment, they have slightly different distributions. So if you look at FGF14, so here's the soma right here. Here's this axon initial segment where the sodium channels are, and you can see this cluster very close to the soma, where with FGF13, it seems to go down further. And we were using a custom antibody right here, so we wanted to validate that these antibodies were actually working the way that they should. And in addition, if you look at the FGF13, you see a lot of FGF13 within the soma itself. So sodium channels, as I mentioned, are mainly concentrated in the axon initial segment. So here is this nominally a sodium channel binding protein sitting in the soma where the concentration of sodium channels is less. And so we wanted to make sure that our antibody was working. And then as I'll show you, we're going to explore what the FGF13 does within the somatic compartment. And so this is just taking GFP tagged versions and showing that FGF14, which is only in the axon, when we tag it, it only goes to the axon, and FGF13, depending on which of those splice variants that I mentioned at the beginning, either S or one called VY, is you see it in the axon, but you also see it within the somatic compartment as well. So it looks like our antibody is accurately reflecting what's going on. So we switched from slices to cultured hippocampal neurons so we could do a little bit more fine-tuning of our imaging, and it's a little embarrassing to kind of show images here. I mean, you walk down the hall, and I think there's like a new fancy microscope in every single corner. Sometimes there's 3 or 4 in every corner. And we don't have that capability at Duke, but we were able to get some nice images. And what we saw in the slices, we could recapitulate with cultured hippocampal neurons. So here is ankyrin G, which is a marker of this axon initial segment where the sodium channels cluster. And you can see that FGF14 is essentially restricted to this proximal portion of the axon in this axon initial segment. And FGF13, however, extends much further. This is quantification of many, many cells, and you can see that there, as you get more distal from the axon, that you have more FGF13. And then in addition, you have all this somatic FGF13 as well. So multiple differences for these two very homologous proteins, both of which are capable of binding sodium channels. This is some super resolution microscopy showing that even within the axon, that these two have limited overlap. And so if you look at FGF13 versus FGF14, you can see that there are limited spots where both are present. They both overlap relatively well with ankyrin G, which is the marker of the axon initial segment, but these two FHFs seem like they're doing different things even in the axon where both are concentrated. So using this knockdown approach, we wanted to query what happens when we knock down either FGF13 or FGF14. So here are hippocampal neurons where we're looking at the sodium channels using an antibody that recognizes all sodium channels, and using ankyrin G to mark where the axon initial segment is. So here's the soma, here's the axon, identified by ankyrin G. And you can see that when we knock down FGF14, is we see a marked reduction in the amount of sodium channels that get out to the axon. In contrast, with FGF13, we see almost no change at all. And here's the quantified data over here. So about a 50% reduction in the amount of sodium channels when we knock down FGF14, and very little change with FGF13. And just looking at ankyrin G, we don't think we're perturbing the axon initial segment itself, because the ankyrin G signal is essentially unchanged in both cases. So even in the context of the axon, where both proteins are present, it seems like they have distinct functions. So we next wanted to see what's going on with the sodium channels when we knock them down. So here is a series of Western blots where we're looking at the biotinylated fraction, or the amount that's on the cell surface. And what we found that was absolutely fascinating and unexpected for us is that when we knock down FGF13, we actually saw more sodium channels at the cell surface. And when we then looked at whole cell current from these neurons, when we knocked down FGF13, we also saw more sodium current. So consistent with more protein getting to the cell surface, we saw more current. This is a summary of that data here, and this is a summary of the knockdown. FGF14, I showed you when we knocked it down, there's less sodium channels in the axon initial segment, and we see less current with FGF14. So that's very consistent with what we saw by imaging. The FGF13 knockdown was quite a surprise to us. We saw more current and more protein at the cell surface. So let me just step back, and if you look at these traces right here, hippocampal neurons have got all these long processes, so they can be a little bit challenging to clamp, and you can see these kind of escape spikes here. And so we decided to take advantage of that by using a protocol that had also originally been described by Bruce Bean, where if you give a pre-pulse, you can essentially get rid of these escape spikes. and you can isolate the somatic versus the axonal currents. And so when we use that pre-pulse protocol, and so here are some of these escape spikes, and you can get rid of them when you do the pre-pulse because you get those channels essentially inactivated. And then you can do a subtraction, and you can separate those channels that are in the soma versus those channels that are in the axon. So let's pay attention to the red traces, which is the FGF13 knockdown. So with the somatic sodium currents, you can see a marked increase after FGF13 knockdown, consistent with more channels at the cell surface and the more current that we saw without the pre-pulse. And you can see that FGF14 reduces knockdown, reduces the number of total sodium channels, as I showed you before by the biotinylation, and also the amount that are in the soma. When we use this protocol now to isolate the axonal compartment, you can see that FGF13 is doing nothing to the axonal sodium channels, consistent with the absence of an effect after knockdown by immunohistochemistry or immunocytochemistry of FGF13. And so this was quite intriguing to us because neurons are polarized. You have this structure called the axon where the sodium channels are highly localized, and how they get localized is quite interesting. And so the current model is that sodium channels are translated and they go everywhere. They go to all compartments, and then they're selectively endocytosed from the somatic compartment, and they essentially get locked in through interactions with ankyrin G to the axon initial segment. So channels go everywhere, and then they get selectively taken up from the soma, and they get selectively locked in to the axon, and that's how you get this concentration of sodium channels in the axon. The mechanism by which sodium channels get selectively endocytosed from the soma is not very clear. And so when we saw this data by knocking down FGF13, that we were seeing more channels, particularly more channels within the soma, we thought that FGF13 could be playing a role in the endocytic process. And so by knocking it down, we're losing some of that selective endocytosis. And let me go over some other data. So using this RA mutation that I showed before, we're able to show that the two processes, getting via FGF14 the sodium channels to go out to the axon and getting the sodium channels to be selectively endocytosed from the soma with FGF13 required direct interactions with the sodium channels. So we have the same R to A mutation as I described before, and this is the same summary data that I showed before, and looking specifically at NAV16 with purified protein, we can show that that RA mutation leads to decreased interaction. And so now if we go back and look at the currents, when we start with the scramble control, and then when we knock down, we don't see that restoration of decreased current. If anything, we see even more. FGF14 is the opposite story. FGF14 is the one that's taking it out to the axons, knockdown, here's scrambled, here's knockdown. Rescue with wild type would go back to the scramble control, but rescue with the R to A mutation is unsuccessful in restoring the currents. So it looks like that you need this direct interaction. So the model that we're currently working on, as I mentioned, is that they get transcribed, they get out to the soma, selectively taken up from the soma, and then concentrated through interactions with ankyrin G in the axon. So we think that these FGF13 molecules, through their direct interactions with sodium channels, are very important for this selective endocytic process. And so we're trying to figure out the specific molecular mechanisms for that currently. So let me change gears a little bit to go back to talk about calcium channels. When we looked in detail with our FGF13 antibody, we saw this striated pattern. And so this, for those of you who are interested in cardiac myocytes and calcium channels, looks like a T-tubular distribution. And so even though these had been originally described as sodium channel interacting proteins, we thought that based on their distribution that they might be doing other things. Now, in addition, in trying to figure out what other things that they could be doing, is we had done a proteomic screen looking at interacting proteins for FGF13. And so one of the proteins that we pulled out is this protein called junctophyllin 2. This is a really interesting molecule in that it has a single transmembrane domain that sits in the sarcoplasmic reticulum and associates with ranitidine receptors. And then it has a long alpha helical domain, and then it has a series of of motifs called Morn domains or Morn motifs that allow it to interact with the intracellular leaflet of the plasma membrane. And the purpose of this protein is thought to essentially buckle up the calcium channels that sit in these specialized invaginations called T-tubules to position them so that when calcium comes in, it can activate the ranitidine receptors, which allow calcium release so you can get excitation, contraction, coupling. And this is actually a locus for certain cardiomyopathies. And so we pulled this out of our proteomic screen. We showed that we could immunoprecipitate the junctophyllin with FGF13. And so then what would be the context of knocking down FGF13 and perturbing this interaction with junctophyllin 2? And so here are a series of calcium traces, the control cells, which were uninfected, the scrambled virus, and then knockdown. You can see that you get reduced current summarized right here after knockdown. So we're losing calcium channel current after FGF13 knockdown in the adult rat ventricular myocytes. With biotinylation, we also showed that there is a decreased number of channels that are getting to the cell surface without a change in the total amount of channels. And so where are they going? So this is, cardiac myocytes can be fun to look at because they have this beautiful striated pattern. This is looking at the ranitidine receptor. This is looking at the pore-forming subunit of the calcium channel. And you can see that you have these beautiful 2-micron spacings that overlap with one another. So the calcium channels and the ranitidine receptors are directly opposed to one another for efficient EC coupling. And then what happens after knockdown is you essentially take a shotgun and just blow these calcium channels away. So they're not falling on these kind of 2-micron spacings. So we quantified this by looking at a number of cells. And so in the wild type, you see this beautiful 2-micron spacing, and you can do Fourier transforms and get a really nice signal in the frequency domain, and it gets completely lost after the knockdown. So we know that we're not just perturbing the T-tubules because when we look at another component of the T-tubules, such as the sodium-calcium exchanger, you can see that it is unperturbed. So it's somehow specific for the calcium channels rather than for the sodium-calcium exchanger. We also used a stain called di-8-NEPS, which is a voltage-sensitive stain that kind of gives you this beautiful images of T-tubules. And I can also tell you that using that technique, that the T-tubules are also unperturbed. So it's a specific localization of the calcium channels. So trying to figure out what the mechanism for this is, we went back to our proteomic screen and tried to figure out what other proteins were associating with the FHFs. And we realized that we had 3 out of 4 proteins called cavins. So we had cavin 1, 2, and 4, which are the only 3 of the 4 that are expressed in cardiac myocytes. Now these cavin proteins are interesting, or at least interesting to us, proteins, in that they're very important in organizing these invaginations of the plasma membrane for these structures called caveoli, which are these very important signaling domains. And there are calcium channels that are specific to the caveoli, and it's thought to be that those calcium channels are particularly sensitive to beta-2 adrenergic signaling and potentially leading to hypertrophic signaling pathways. And so here is sucrose density gradient experiments showing that, in fact, that FGF13 is associated in these lipid-rich domains, which is where the caveoli, and you can see this by the caveolin 3, which is a marker of caveoli, where that signals, and that there are calcium channels and sodium channels and junctophyllin 2 that are also in these specialized domains. I also want to point out that if you normalize the amount of protein in these specific domains to the total amount of protein, even though it looks like that this band is just as dark as that band, there's so much less protein in the caveoli that this turns out to be a high concentration of these proteins within caveoli. So we think that caveoli are a very important home for these FHFs, and that they could be organizing signaling within these compartments. And so going back to our knockdown strategy, what we saw after knockdown is that we see more of proteins such as junctophyllin 2, such as the calcium channel, moving from these heavy fraction to these caveolar light fractions after FGF13 knockdown, consistent with what we saw by immunocytochemistry. And here's some quantification of that as well. As you can see, that we not only see more junctifilin 2 and caveolin 3 expressed in these cells, and obviously less FGF13 because that was our target of the knockout, but we also see more in the lipid-rich fraction, which are identified by the caveolin 3 as most likely the caveoli. So what's the consequence of this? So I mentioned before that the caveoli are thought to be specialized signaling compartments. There's work from Steve Hauser as well as from others that suggests that, and you guys have done also some really nice work on this, that suggests that the beta-2 adrenergic signaling pathway, and Tim Camp, I should also mention, was one of the pioneers in this, that the beta-2 adrenergic receptor is also localized in the caveoli, and you can get specific activation of calcium channels within caveoli because of this association. So we thought that if by knocking down FGF13 we're seeing a higher concentration of calcium channels within caveoli, we may be able to see a potentiation of the beta-2 adrenergic activation of calcium channels after knockdown. And in fact, that's exactly what we see. So if you look at the global calcium currents, you can see that you get a reduction in the amount of calcium channels after knockdown, as I already showed you before, quantification over here. But now when we stimulate specifically the beta-2 adrenergic receptors, is you can see a larger potentiation after knockdown than in the scramble controls. And so here are some of those diary plots over here. So it's suggested that, in fact, that this signaling pathway could be revved up after knockdown. And so if that's the case, we thought that this could be a signaling pathway leading to cardiac hypertrophy. So one of the genes that's activated, or one of the transcription pathways that's activated in cardiac hypertrophy is the NFAT signaling pathway. NFAT is a transcription factor that, in the presence of calcium, gets dephosphorylated, moves from the cytoplasm to the nucleus. And so using a GFP-tagged NFAT that we got from Steve Hauser's group, we could then stimulate either control cardiac myocytes or myocytes in which we've knocked down FGF13, and then look at the rate of NFAT translocation to the nucleus. And you can see that we see a faster rate of translocation after knockdown, consistent with these calcium channels localized to the caveoli in those cardiac myocytes. So to just summarize this part, it looks like the FHFs and cardiac myocytes are regulating sodium channels. They also seem to be regulating sodium channel kinetics, and they're also regulating calcium channel trafficking and calcium channel signaling. They don't regulate calcium channel kinetics, and we think that's because, at least so far, we have not been able to identify direct interaction. We think it's more through that junctifilin. And in the context of the knockdown or mutations, that we think that this is a Brugada syndrome locus. So let me see if I can whip through this in the last few minutes and talk a little bit about Calmodulin. This has been a protein of interest in the lab for many years. It is the protein that essentially translates calcium or electrical activities into a calcium signal with inside cells. It's a highly abundant protein. It can be up to 1% in some cells. It binds 4 moles of calcium, shown in the yellow dots right here, per mole of Calmodulin. Binds to all sorts of channels and regulates their function, and that's why we're very interested in it. So here is the C-terminus of the sodium channel that I already showed you before. It had been previously shown that the Calmodulin bound to this particular motif called an IQ motif because it's focused on this isoleucine and glutamine residues right here. And what I'm showing you is a series of mutations that have been identified in long QT syndrome or Brugada syndrome in the cardiac sodium channel, or in epilepsy in the neuronal NAV1.2 channel, or in blue, these are epilepsy mutations in NAV1.1, which is another channel. And you can see that this Calmodulin interaction site is a hotspot. So we focused before on this interaction, so let me move down the molecule and focus now on the Calmodulin interaction. We were able to show that the Calmodulin, in fact, does bind to the isoleucine and glutamine of the IQ motif, shown in blue right here. And if we focus in on the consequences of this, is we had actually done these experiments way before we had gotten the crystal structures. When you knock out the IQ motifs and you completely lose binding, so here's some more ITC between the calmodulin now and the sodium channel, the IQ-AA mutation, no binding whatsoever, the consequence is that you have increased late current, which, as I mentioned before, is a marker for arrhythmias and what causes long QT syndrome. And so what's happening here is you have this abnormal late current. It prolongs the action potential. It leads to increased calcium influx through activation of the sodium-calcium exchanger and arrhythmias. So that structure was in the presence of calcium. The absence of calcium. So we recently got another structure in the presence of calcium. And so the consequences now are that the calmodulin, which was binding in its apo or calcium-free form with the C lobe, the N lobe now comes along and binds to this distal portion of the IQ motif. What's interesting is that the C lobe stays uncalcified. Calcium only binds to the N lobe and then kind of swings around. So it's almost as if the calmodulin is a resident calcium sensor waiting there for calcium to come along and then change this interaction right here. And so if we focus in on that, you can once again, you can see this N lobe become calcified and swing and bind to this distal portion of the C terminus of the sodium channel. Is this important? We think it is. It had previously been demonstrated that there was a Brugada syndrome mutation in this distal portion of the alpha helix of the sodium channel that had calcium sensitivity to the sodium currents, but the mechanism was not clear because everyone had focused up here on the IQ motif. So when we saw this changes in the structure, we thought that we could explain the calcium sensitivity by this Brugada syndrome mutation, which they had shown was responsible, or calmodulin was responsible for by this mutation here. And once again, using the ITC, we could see that this mutation causes about a threefold reduction in affinity. So we think that that calcium-dependent movement of calmodulin is important for regulating sodium channel kinetics, and that's what's going on in this Brugada syndrome mutation. Last few moments, this is not just sodium channel disorders are not just in the heart. So sodium channels are one of the few loci that has been recurrently found as a locus for autism in multiple studies over the last few years. And so there was one particular mutation in SCN2A that we found very interesting, and this was in a family of patients with autism, and it was this arginine 1902C mutation right here. Now, this was of interest to us because it doesn't directly interact with calmodulin, but through a relay interacts with this glutamate residue here that then interacts with this critical lysine in calmodulin. And so we queried what happened when we mutated that arginine to the disease cysteine, and what you see is that in the presence of calcium, for the mutant, you get this gain-of-function shift in activation, which doesn't happen in the wild type, and you see this gain-of-function shift in steady-state inactivation. So it's going to, in theory, change the excitability of these cells in the presence of calcium. Now, I said that this is not a direct interaction with calmodulin, so if we were correct in our hypothesis is that these changes in kinetics are due to this relay of the interaction with calmodulin, then we predicted that if we mutated this aspartate, that we would phenocopy that same mutation. And sure enough, we see that. When we mutate the aspartate, we again see this gain-of-function shift in activation kinetics and this gain-of-function shift in inactivation kinetics. So we believe that the biophysical defect in these patients, this family that has this particular arginine mutation, is because of a shift in the excitability state of these neurons because of the shifts in kinetics of the sodium channel. So I think I went through a lot, and I probably went a little bit over, but hopefully I was able to give you a sense of two different accessory subunits, these new sodium channel and calcium channel chips that are regulating sodium channels and calcium channels, doing various things both in the brain and the heart, and leading to different diseases both in the brain and the heart. I just wanted to show one last slide. This is really not part of the talk, but we looked at a patient that came from this very institution. We published a year ago, and Joelle Miller, who's a pediatric cardiologist here, sent us this patient, sent actually Ron Cantor, who's a pediatric cardiologist that used to be at Duke, and we studied this patient, and we found out why this patient had a particular defect in their calcium channels that was leading to the arrhythmias. Not part of this talk, but it's my VTCRI connection, so I wanted to share that with everyone. So with that, I wanted to just point out the people that have done this work. So I mentioned Juan Pablo's recent work that we just submitted. Xiaojun Wang was the driving force behind the structural work. Eric Wei is the person who's doing that caveolar experiments in cardiac myocytes. Henggang Wang and Haidun Yan did some of the electrophysiology for the heterolog expression systems and the neurons. Jessica Hennessy is a former MD PhD student who started some of the FHF work along with Chuan Wang in the cardiac myocytes. And here are the people that have been kind enough to give us money. So thanks for your attention. I'd be happy to answer any questions.
So really interested by the FHFs going in the nucleus. And also like a lot of ion channel C-termini can be found in nuclear localizations. Have you looked at anything like the complexes going up there and the relationship there?
Yeah, so we're intrigued by that too. When they were first described, there is a particular isoform of FGF13 that seems to have an NLS. And so when they're first described 15 years ago and they made GFP tagged versions, people saw them going to the nucleus. We see that as well. It's one particular isoform. It's not the one that's dominant in cardiac myocytes. It's more important in the brain. I didn't show you that that's not the isoform that's involved in the endocytosis. So if we rescue with the NLS version, it doesn't do anything. It doesn't work. Only this other one that stays out in the soma. So yeah, what does it do in the nucleus? I mean, we would love to know. We have all sorts of weird things that we see in our proteomic screen that we don't know whether they're garbage or they're giving us some interesting signal, and we just haven't had a chance to explore that yet. So I don't know whether they're going along with the C-terminus of the voltage-gated sodium channels and activating transcription, but you know, more grants, hopefully, more projects. Yeah. Yeah.
I have a question about the cerebellar-Purkinje cells.
Yeah.
So in the FGF14 mutants, did you ever look to see if the beta-4, sodium channel beta-4 expression was altered? Or do you think that maybe beta-4 is not really needed for that open channel block?
Yeah, so that's a good question. So it's been very hard to actually pin down whether the beta-4 C-terminus is really critical or not. So, I mean, there's beautiful work that if you knock it down, that you can see a reduction in the resurgent current, but you can't recapitulate that in a heterologous expression system. So our interpretation was that beta-4 was not sufficient, and we actually, one of the reasons we got into the project is if you look at the N-terminus of the FGF14, that it has a lot of the same properties, these positively charged arginine and lysines, as the beta-4 subunit. And so I didn't show you, when we chop off the N-terminus of the beta-4 subunit, we also see effects on the resurgent current. So we're just setting up experiments like that now to see if we can recapitulate this in a simplified system. Your particular question is a good one, which we hadn't thought of. Did we somehow affect the presence of beta-4? And so I don't know. It's a good thing for us to look at. So we did. So when we knock down FGF14 and we look at the granule cell to Purkinje cell synapse, we see less calcium channels and less calcium influx in the presynaptic boutons. And so we think that the disease process is actually quite complicated, that it's probably a combination of defects in the granule cell and defects in the Purkinje neuron. At the granule cell, probably at the granule cell to Purkinje cell synapse, in terms of effects on the calcium currents. There's decreased calcium currents, decreased calcium influx. And in the Purkinje cell, it's this change in sodium channel kinetics, so you get this change in firing. So we have looked at that. We've reported that as well. Yeah.
Looks like in the sodium channel, both Calmodulin and FDA defining to the phase very close. I mean, if it's possible, there are
So there is one critical place where all three molecules, the sodium channel, the FHF, and the Calmodulin, all interact. And we have played around with mutating the three different residues, and we definitely see effects. So we're now finishing up. So we're very interested in late current. I kind of went by it quickly for the neurons, but in cardiac myocytes, that's absolutely critical because that's what drives long QT syndrome. The mechanism at the molecular level of late current is really not well understood. There are some mutations in the 3,4 linker that appear to be really important because that's the lid that kind of is closing the door or closing the channel. But there are mutations that are scattered in the C-terminus that have nothing to do with the 3,4 linker. And they seem to cluster around the Calmodulin and the FHF area. And so we now have looked very carefully at the mutations at a structural level and at a functional level, and we think that they cooperate to regulate the late current. And so we're trying to kind of finish up and really get an understanding of exactly how they interact. So I can explain it in a little bit more detail, but it takes a long time.
Go ahead, Alex.
Yeah.
Are there any links between this FHF, the function is altered? Is there any physiological mechanism where the system will be used to adjust function of the channel?
So I think there probably are. So we've identified the FHFs, and other people have also seen this as phosphoproteins. We believe that phosphorylation will change the affinity of interaction with the channels. And then, this is always the kind of, I think, the way that science develops. I mean, so they were cloned as these FGF-like molecules. People work on them. It turns out they have nothing to do with FGFs because they don't get out of the cell. They don't signal. Someone finds that they're the locus for spinocerebellar ataxia. Someone else finds that they're sodium channel modulators. And so everyone focuses on that. And then, because we can't explain some of our data, start kind of pushing in other areas. And I can tell you that every time we look, we find different things. So that's a long answer to, I'm sure that they're probably involved in signal transduction pathways. There's one report of a USE2 hybrid screen that suggests it binds to a component of the MAP kinase signaling pathway. So I think that they probably are very rich in their ability to regulate multiple processes, and hopefully we'll be able to explore it.
So in the autism story, is there any link between the heart and the brain? We have a scientist at Virginia Tech, Martha Bell, who studies, I think, autistic infants, and she finds this change in heart rate variability in response to stimuli in kids that show early signs of autism. Maybe some of these channels are expressed earlier in development.
Yeah, I mean, that's very interesting. I wasn't aware of those data. There is a connection between epilepsy, which is also a big, ion channelopathy, and cardiac defects in terms of the most common cause of patients dying from epilepsy is a sudden cardiac death, something called SUDEP, or sudden unexpected death in epilepsy. And so we've kind of dabbled in this area, and we think that some of these accessory subunits are regulating an ion channel in the brain, also regulating an ion channel in heart, and it can manifest as an arrhythmia of the brain or epilepsy versus an arrhythmia of the heart. And there's some data from other groups that support that as well, but I don't know about the autism. I need to look that up.
So I'm just curious, have you looked at FGF14 impact on the alveolar endocytosis? Does it show differing impacts of using 13 versus 14 and trafficking to the fisher segment?
Yeah, so that's an interesting question. So there's caveolin expressed in neurons. From what I understand, no one has been able to demonstrate that there are caveoli in neurons. And so FGF14 doesn't appear to affect the somatic channels, where FGF13 does. And we've done experiments with dinosaur, and we're now trying to do some other labeling experiments to really kind of tease apart that process. So how that endocytosis actually works is unclear, but FGF14 doesn't appear to be involved in that. So we knock that down. We don't do anything to the endocytic process. We think it's more somehow trafficking or targeting the channels to get to the ankyrin G-rich axon initial segment.
Okay, well, thanks so much for a wonderful session.
Thanks.